Hey, Bio30s, and welcome to our final discussion in the molecular genetics unit, genetic technologies. I want to discuss today a specific branch of biotechnology, and biotechnologies refer to any technology that basically are technologies that are based on biological structures or biological systems. And we've talked about biotechnologies before, most notably in Bio20 vaccines. Today, I want to discuss the specific branch of biotechnologies known as genetic engineering, which basically humans are basically changing the genetic makeup of organisms and how they do that. Um, Today's lesson, there is no homework for it. There's a lot of videos that are going to be referenced in this video. I would ask you to try to watch as many of them as you can and just make sure you're taking some good notes today because there is no homework assignment per se, but you are going to be responsible for knowing this material and you will have a chance to kind of test your knowledge on this later in the unit when we get to our GMO position paper that you'll be doing as sort of your final project coming up. So, anyways, genetic engineering sounds like it's a very brand new field of science, but really that couldn't be anywhere. I guess I can't even say it. Uh, it's a lie. That's how I'm going to say it. It's a lie. Genetic engineering has been around for thousands of years. Artificial selection has been used by humans to manipulate crops, to manipulate organisms, breeds of dogs. You know, broccoli, cauliflower, kale. These all originated from wild mustard plants that were selectively bred by humans. Now, selective breeding, artificial selection takes a long time, and now that we know more about specifics of DNA, we can go in and surgically basically pop out a gene of an organism and put it into another one if we want. So that has sort of brought this technology more to the forefront and has sped up the process. But when we speak about genetic engineering, it has been around for thousands of years. It's just the way that we go about it now over the last 50 to 60 years has very much escalated and accelerated. And the process by which we can manipulate organisms because of these new technologies has also accelerated. So we're not dealing with having to breed generation after generation after generation to get an organism to do what we want it to do. And even more than that, we can pick and choose genetic information from different organisms. Because remember, we talked about in our protein synthesis lesson that DNA is universal. It's the same nucleotides, ATGC, that run through every DNA of every organism on the planet. So we can basically transplant DNA from one organism into another, which would be creating what we call recombinant DNA, which I'll get to here right away, and eventually genetically modified organisms or transgenic organisms. So, and again, like I just mentioned, the key to all of this genetic engineering, at least now, is creating recombinant DNA. And recombinant DNA, by definition, is essentially DNA that is basically created by combining the genes or the DNA of at least two different organisms, and in some cases it can be multiple organisms that are basically combining DNA together, or that we're combining the DNA of together to create a genetically modified organism. Now, how do we do this? Well, in order to create recombinant DNA, it's kind of simple. If you've got a piece of DNA and I want to pull out one little gene off a piece of chromatin or chromosome, I want to get rid of that gene. I'm going to have to cut that gene out to extract it out of that piece of DNA. So I need essentially a fancy pair of scissors. And we don't really have microscopic scissors that can do it. So what we do instead is we use essentially what are called, now back in the day we used something called restriction enzymes. And these enzymes are produced by bacteria, and they're essentially a form of immune response to invading bacteriophages, which are viruses that actually invade bacteria. So these enzymes, these endonucleases, and one of them would be EcoR1 is a very classic example of such an enzyme. These enzymes are produced by the bacteria so that when the genetic information is injected from a virus into the bacteria can actually break apart the virus DNA before it can actually hijack the cell of the bacteria to start producing copies of itself. So back in the 1960s, this was invented back in about 1968 by a scientist by the name of Hamilton Smith. this technique was used for decades to basically cut pieces of DNA so we could extract certain genes, and then we could take those genes, we could cut another piece of DNA in another organism using restriction enzymes, and then basically we could take that gene from the one piece of DNA and stick it into the middle of another piece of DNA from another organism. So we would have our scissors, which are our restriction enzymes, but then we would also need something to basically put what were called these restriction fragments, these little pieces of cut DNA that we cut using these restriction enzymes are referred to as restriction fragments. They also have like little overhanging overlaps with their nucleotides called sticky ends that basically will allow two restriction fragments to then basically overlap each other and then get sealed back together again. But you need to be able to glue these pieces of restriction enzymes back together again. So you need essentially a glue. Well, we already know about a glue. We know about an enzyme that's produced in cells all the time during DNA replication to basically put RNA primers and stick them back together to sections of DNA, or on your lagging strands, your Okazaki fragments that have to be all glued back together again. We've talked about DNA ligase. So we can use DNA ligase. After we've cut the DNA with these restriction enzymes, we can then put the restriction fragments into another piece of DNA and then essentially put Humpty Dumpty back together again using a DNA ligase, the enzyme DNA ligase, to do that. And then once we've done that, now we've combined, if the genes from this piece of DNA and this piece of DNA over here are from two different organisms, well, now we've created what's called a transgenic, or a genetically modified, or a GMO organism, or a GMO. And that is basically an organism that has multiple pieces of DNA from different organisms in it. And I'll give you a couple of examples, or right now I'll give you an example. Golden rice is a great example. Golden rice is a GMO version of rice that has DNA from 4 different species in it. It has DNA from wild rice, daffodils, a species of fungus, and a species of bean, as you can see here on this slide. And it will go into details to what each of the genes are that are being added and what function they provide. But essentially, putting the genes of these 4 different organisms together has increased the nutritional yield of the rice. So that is kind of why it's been done. So that would be an example of a case where we've basically split DNA apart, found specific genes that we want, and then combined them together into one section of DNA that then becomes essentially our GMO, our recombinant DNA, which will then lead to the formation of our GMO. There is, and by the way, one of the first GMOs out there, very important one, because sometimes GMOs get bad raps, but one of the earliest GMOs that was actually out there was in 1982. They invented or designed, I guess, a bacteria that could produce human insulin, which has been huge for diabetics, which goes back to our endocrine unit from earlier this semester. So if you think GMOs are always bad, and again, you will be doing some research on this when you do your GMO project, but GMOs may have gotten maybe a bit of a bad rap, which they don't deserve. They're not safe. In some cases, these GMOs have been real lifesavers. For people that are diabetic, these GMO bacteria that can produce insulin has been a very cheap and easy way for us to produce insulin on a mass scale for people that have type 1 diabetes that require insulin shots on a daily basis. So that's something to keep in mind. Before I get off the topic of recombinant DNA, well, I'm going to be kind of referring to it throughout today's lesson. There is a newer way to create a recombinant DNA, and this is a version or a method known as CRISPR-Cas9. CRISPR-Cas9 is also it's also based on the bacterial immune system, but it is a much more efficient and more targeted approach to finding specific nucleotide sequences in a piece of DNA. It was developed by two female geneticists. One of them is named Jennifer Doudna, and the other one is, Emmanuelle Charpentier. You'll probably see them on my walls in my classroom. They're usually one or both of them are up on the walls at any given time, and they are really rock stars in the field of genetics right now. And anyways, if you're interested about how CRISPR-Cas9 works, short video here that will explain a little bit of the the stuff behind the CRISPR-Cas9 effect. And this, if you, in case you're interested, you wouldn't be expected to know specifics about CRISPR-Cas9, other than you should know that CRISPR-Cas9 is a very specific and more accurate way to cut DNA in order to create these fragments of DNA that can then be inserted into recombinant DNA. So we can use CRISPR-Cas9 to excise genes that we want and then cut apart pieces of DNA that we wish to insert those genes into. And I'm going to get back to it in a second, but I will get back to another topic based on CRISPR-Cas9 in a couple of minutes, a very controversial issue that you might read about here on the slide, but I'm going to refer to it in a few minutes. Okay, here's a little slide that kind of shows you a diagram of sort of what CRISPR-Cas9 sort of does. The video, I think, on the previous, the one that I alluded to earlier, the one if you watch it, gives you a much better idea of what CRISPR-Cas9 actually does. So anyways, getting back to GMOs before I get off the official topic of GMOs. GMOs, the first GMO ever was in 1973. It was a bacteria. First, the vertebrate GMO was a mouse in 1974. Genetically modified food has been sold since 1994, and it's been raising a lot of questions. And again, you're going to have some time to discuss that in your position paper. Some of the things you want to maybe discuss in that position paper, should someone be able to own an organism? Can we patent? Because that's what's happening now. These GMOs are being patented by corporations that actually own these organisms. And can anyone actually own an organism? Well, that's an ethical debate that needs to be addressed. Again, can you patent an organism that, may or may not be something that people should be, doing, but people right now do. You can patent an organism. And as students wanting to learn more, what are some of the challenges studying the effects of genetically engineered food products? So one of the things you have to watch out for when you're doing, any kind of research on GMOs, you got to make sure that you're getting accurate sources. So anytime you're doing research for biology in general, you should be using Google Scholar. You shouldn't just be doing Google searches regularly. You should be going to the web engine or the web search, Google Scholar. That's going to give you scientifically, validated papers and websites that basically are basing it on science, not on necessarily crackpot theories that GMOs are giving us all third eyes in the middle of our forehead. Some crackpot in their basement can put that on the internet and people believe it. We've had this discussion before when it came to vaccines and the whole vaccine debate. This is also a very contentious area in science right now about genetically modifying organisms, and there are obviously pros and cons, but you want to be able to get the real science behind it, not crackpot theories that are not based on good scientific practice. So keep that in mind. Again, another slide here with some information about GMOs that you might want to have as a starting point before you get into your position paper on GMOs. And again, a little bit of humor there. The GMO rabbits that are taking over. It's not Planet of the Apes, Planet of the Hares. Okay. That's why I'm not a comedian. That's why I'm teaching it in my basement right now. Yeah, okay. Anyways, getting back to our topic. Some other things that we can use for genetic technologies or genetic engineering. One of them that's been had a lot more positive, spin in the news and on the web is a technique known as gene therapy. Now, gene therapy is basically a way to quote unquote correct a cell or a group of cells that have a disorder. For example, this can be used for patients with cystic fibrosis. And cystic fibrosis we talked about in Bio 20. It's a disorder that produces more than normal, above average amounts of mucus in the respiratory system and in the digestive system, which causes problems with breathing and with digestion, respectively. What we can do through an inhaler is we can actually introduce into the lungs what we call a DNA vector. And a DNA vector is a method of delivery for DNA that it's going to correct the problem. We are going to target the gene in those cells in the respiratory system that have a faulty gene that has the cystic fibrosis gene, and we're going to try to inject those cells using a DNA vector to basically replace the defective gene and replace it with a good one. And usually DNA vectors, when they're used, are viruses. Viruses that have been basically reprogrammed, that have been injected with human DNA rather than viral DNA to basically do what viruses do, and that's to infect cells to replace DNA in the cell. And in this case, they're going to carry genetic material to a host cell, in this case in the lungs, to sort of replace them. Now, what I've just described to you is one form of gene therapy known as somatic gene therapy. That's replacing defective genes in body cells to basically fix a potential problem, like in this case, cystic fibrosis. There is a more controversial form of gene therapy, which is technically illegal, but scientifically it can be done, and it has been done. And if you want to see the results of that, and it's scary stuff that you may have heard about, that scientist in China who basically experimented and actually created designer embryos. He basically modified the DNA of the zygotes and basically tried to make those their DNA more resistant to the HIV. And he did that experiment, and there was a lot of controversy because technically you shouldn't be doing gene therapy on germline mutation or germline therapy. You shouldn't be doing this with gametes, eggs, sperm, or zygotes, because this is a very slippery slope. This becomes a, almost like the Aryan race, right? I mean, this has happened in world history in the 20th century when the Nazis came to power. They wanted the master race, and this is very, very scary stuff if it's not proceeded with very carefully, because it's the whole, can we make designer babies? Can we make, babies to look and be a certain way? And that is a very ethically dangerous area to be in. And for now, scientists around the world have agreed that this is science that we shouldn't be conducting. Yet the scientist in China did it very recently, and he sort of kind of disappeared after this all happened. It was like he was all in the news, and then all of a sudden sort of disappeared. The Chinese government just sort of kind of put him aside, and I don't even know what happened to him. But I mean, it's an interesting story about what he did. So take a look at that link if you haven't seen it. Some scary stuff. Somatic gene therapy, though, is quite legal, and we are doing it to try to fix somatic disorders in specific parts of the body, whether it's the respiratory system or the digestive system or other parts of the body. And we're generally going to use stem cells for this. There's a lot of use of stem cells, and you may have heard about stem cells, and we'll discuss more about stem cells later in the reproductive unit. But right now, if you want to take a quick peek about what stem cells are and what their link is to all of this, you might want to give that video a quick watch over there about stem cells and what they're doing and what's the big deal about stem cells. But I will get back to stem cells when we get to our next unit on reproduction, when we talk about prenatal development. A couple of other forms of DNA vectors that can be used. These are kind of cool ones because they're a little different than what we would use in humans. These are DNA vectors that are used in plants. So there's one called agrobacterium-mediated transformation. In that case, the DNA vector is not a virus. It's a bacteria that actually acts as the gene vector and then alters plant DNA. So same idea, it's using a DNA vector, but in this case, it's not a virus. We're using a bacterium to actually invade the cells of the plant to actually add the DNA to the plant that we want to add. And this could also be used in not just gene therapy, but also to perhaps introduce genes from other organisms. So this could also be used as a way to create recombinant DNA as well. And one other method, which is just great because I love the name of it, there's also something called a gene gun. And it's just like the name implies, it's actually a gun that fires DNA into plant cells, and then we can take those cells and try to grow them in a culture, and then basically try to see if the DNA from the gene gun has actually entered the cells and has actually modified the DNA of the cell. Not very accurate, but it's a really cool method. And if you want to see what a gene gun looks like and how it works, watch this video right here. They're going to walk you through a gene gun and how it actually works. All right, so we've talked about gene therapy now. We've talked about recombinant DNA and creating GMOs. There's a couple of other things that we need to be able to do. We don't want to just be able to introduce DNA into a cell using like a DNA vector. We don't want to just be able to cut the DNA like we do with restriction enzymes or CRISPR-Cas9. We don't just have to be able to put the DNA and glue it back together again if we're using something like DNA ligase. There's a couple of other things that we need to be able to do with genetic engineering. One of them is to actually locate the genes that we need to put into the organism. If there's a gene that we need to fix, like in gene therapy, like the gene that causes cystic fibrosis, or if we're trying to isolate a gene, let's say in golden rice, that we want to add from the fungus that's going to give it a specific trait that we want from that fungus, you first have to find the gene. So if you know that the gene is on chromosome, 27, you still have to know where on the chromosome to cut. So in order to find the genes that we want to actually use for gene therapy or to make recombinant DNA, we need to use something called the DNA microarray. And a DNA microarray is just a simple way for us to locate specific genes on chromosomes. And we do this by basically using mRNA, which we talked about in protein synthesis, that it's going to copy the DNA, and we're going to use the mRNA to make a copy of the DNA, which we can then react with the regular strand of DNA, and then through that interaction, we can isolate specific genes. And that's essentially what a DNA microarray does. Kind of a very brief overview. More details, if you want, are on the slide. But that's pretty much what you need to know about a DNA microarray. What about if I want to do these experiments, but I only have one piece of DNA? That doesn't really help me. I need to be able to find ways to quickly duplicate DNA, and we know that DNA can duplicate itself. So what we do is we use a process called a polymerase chain reaction, which was basically developed in 1983 by a guy by the name of Kary Mullis, who is another big rock star in the field of genetics. Basically created this technique, which basically creates a chain reaction of DNA polymerase reactions, which basically copies one piece or a few pieces of DNA into millions of pieces of DNA in a very, very short time. So we can use this process to actually copy DNA. So we've got all sorts of methods that we need to be worried about in genetic engineering. Cutting DNA is going to be your restriction enzymes and your CRISPR-Cas9. Gluing DNA back together again is going to be your DNA ligase. DNA microarrays are going to allow you to isolate DNA, the specific genes that we want to use for our genetic engineering. And the DNA polymerase reactions are going to be used to copy that DNA that we need in order to make these reactions happen. There is one last technique that I'd like to talk to you about, which is used to identify individuals. So this isn't a technique that's used to modify DNA or to change DNA at all. It's just to find out the connection between a piece of DNA. This is quite often used by forensic investigators. They find DNA at a crime scene. They want to isolate the DNA from a crime scene. They want to see what that DNA looks like. Maybe you may have heard of stories of soldiers that are found in battlefields 100 years. They're still digging up, unfortunately, some of these soldiers that die in World War I. They sometimes will find remains of soldiers even 100 years after the battles that basically died on the battlefield and were basically they were never properly buried or given a proper funeral. They can take the DNA from these soldiers and see if they can identify those soldiers based on relatives that are living back in Canada or in England or in France that may have been the descendants of that soldier. So this technique is good for all of this. It can also be used for a paternal to determine the father of a baby or a mother of a baby. Again, the mother of the baby, but let's say it's an adopted baby, and the child wants to know who his biological mother or father is. They can do one of these to determine the DNA if they don't have records of who their mother or father is. The process I'm referring to is a process called gel electrophoresis, which essentially does a process called DNA fingerprinting. And what this process is, and there's a whole slide about it here, and you can watch the video right up here about gel electrophoresis up here just to see what the process entails. But essentially, DNA is a negatively charged molecule. We're going to use electrical fields to basically cause DNA to migrate through a gel to a positive electrode. And depending on the size and the weight and the the makeup of the DNA in question, the different fragments or sections of DNA are going to move at different rates because they're going to if they're bigger, they're not going to move as much. If they're smaller, they're going to move more. And when we do this through this gel, what we will get is we will get a distinctive pattern that you can see here. And that pattern is distinctive because of the nucleotide sequences in that section of DNA. And because everyone's DNA is unique, unless of course you have an identical twin, that pattern is going to be unique, and there will be DNA or genetic markers in that fingerprint that might lead a scientist or a forensic investigator to determine, maybe if it isn't that particular individual, that the DNA that we're comparing it to might be a relative or a close relative or a relative in the family. That's how we would be able to figure out paternal-maternal connection between the child, or a fallen soldier on the battlefield, whether or not that soldier is in fact the relative of these people living in another country, because we think it's a soldier, but we're going to compare the DNA of that soldier to the soldier's descendants living back in Canada or in Britain or wherever, to see if that actual DNA is a match to that family, so you can actually determine who the fallen or the unknown soldier is in the battlefield. And this is sort of what the readout looks like when you're done this process, and that pattern is unique like a fingerprint. That's why we call it DNA fingerprinting, and that's why it works very, very well. Another example here, just showing you sort of the base pair sequences there. Take a look at this diagram here. Here's an example of how it would be used. We've got my child's DNA fingerprint over here. I've got parents A and parents B, parents C, parents D. Which of those parents, is it parents A, parents B, parents C, or parents D, that are the possible parents of the offspring? So what I'm looking for now are genetic markers between the two parents that match the genetic markers of the child. And if I look, the only parents that have all the genetic markers of this child over here between the mother and the father are going to be parents B. So take a look at parents B and see why they're the parents of the offspring rather than parents C, D, or A. That's sort of how a DNA fingerprint kind of works. And well, what's next? I mean, the sky's the limit with genetic engineering. Take a look. You've got food and livestock modification that's ongoing. You've got gene drives, gene therapy, human germline experiments like you saw in the video earlier. That's a whole, are we going to go there? Are we not going to go there? Hopefully right now we're not going to go there because we're not ready for this in any way, shape, or form. And then, continuing design of organisms. If you want to sort of see what the future has in store, take a look at the video there. There is really no homework for this. Again, you're going to have your position paper coming up right away. But please be familiar with all the processes that I've mentioned here today, whether it's CRISPR-Cas9 restriction enzymes, the use of DNA ligase, what gene therapy is, gene vectors, polymerase chain reactions, DNA microarrays, restriction fragments, recombinant DNA, GMOs, and of course the last process we talked about, gel electrophoresis and DNA fingerprinting. That's it for molecular genetics. Guys and gals, we're done. Congratulations, you made it through another unit. Join me again next time, and we'll be starting our next unit, which will be on human reproduction. Until then, have a great day.